Somewhere in Chicago, there's a freezer full of donated human brains that may hold one of the most important clues we have ever found about aging. Some of those brains belong to people who died in their 80s and 90s with memories as sharp as someone 40 years younger. Others belong to people the same age whose minds had already faded years before death. For decades, scientists assumed the difference between those two outcomes came down mostly to luck, lifestyle, or simply which diseases happened to find you first. A study published in February 2026 in the journal Nature suggests something far more specific and far more biological is actually going on, and it starts inside a part of the brain barely the size of a thumb. This is the story of the superagers, a small, extraordinary group of people whose brains appear to be quietly rewriting the rules of what growing old is supposed to look like, and the genetic difference researchers just discovered that may explain why. The term superager did not originate in a lab coat moment of scientific inspiration. It was coined in the late 1990s by Dr. Marcel Masulim, a neurologist at Northwestern University, who noticed something strange happening among a small subset of his older patients. By strict definition, a superager is someone 80 years old or older whose memory performance, measured through standardized word recall testing, matches or beats people 2 to 3 decades younger. Not slightly better for their age. Better than people who are, in some cases, still raising teenagers while the superager is celebrating a 9th decade of life. Northwestern's superaging program has spent more than 25 years tracking these individuals, collecting everything from psychological questionnaires to, eventually, their donated brain tissue after death. Over that quarter century, a picture has slowly emerged. Superagers are frequently described as unusually social and extroverted. Their brains tend to shrink more slowly than their peers. Some carry the same hallmark proteins associated with Alzheimer's disease, the sticky plaques and tangled fibers that typically choke off healthy brain function, yet somehow remain cognitively unharmed by them. Something about their neurological wiring appears to be different at a fundamental level, and until recently, nobody could say precisely what that something actually was. The 2026 study, led by neuroscientist Orly Lazaroff at the University of Illinois College of Medicine in Chicago, set out to answer that question directly by focusing on one specific brain structure, the hippocampus, the small, seahorse-shaped region tucked deep within the brain that plays a central role in forming and retrieving memories. Lazaroff's team examined donated brain tissue from 38 individuals spanning 5 distinct groups, ranging from healthy younger adults to typical older adults, to people in the earliest stages of cognitive decline, to people who had died with Alzheimer's disease, to 6 precious superager brains supplied by Northwestern's long-running program. Using detailed molecular analysis capable of examining individual cells within the tissue, researchers went looking for something specific: neurogenesis, the process by which the brain generates brand new neurons. For much of the 20th century, scientists assumed adult mammalian brains, including human ones, were essentially fixed at birth and capable of producing meaningful numbers of new neurons once early development was complete. That assumption began to crumble once researchers documented ongoing neurogenesis first in rodents, then in primates during the 1960s and 70s. Evidence for the same process occurring in adult human brains has trickled in more slowly since, concentrated specifically within a small subregion of the hippocampus called the dentate gyrus, though the overall picture had remained frustratingly inconsistent from one study to the next. What Lazaroff's team found settled a meaningful piece of that long-running uncertainty, and did so with a genuinely striking result. Superager brains contained roughly twice as many newly formed immature neurons as typical healthy older adult brains, and about 2.5 times as many compared with brains from people who had Alzheimer's disease. In other words, while the typical aging brain slows its production of fresh neural material with each passing decade, superager brains appear to keep the factory running at something closer to full capacity, deep into old age. It would be reasonable at this point to assume this is simply a story about neuron counts, interesting but not necessarily genetic in nature. That assumption would miss the actual significance of the finding. According to Northwestern's superaging program, this research represents the first documented discovery of an underlying genetic difference separating superagers from typical older adults. Not merely a difference in outcome, but a difference in the underlying molecular machinery producing that outcome. Lazaroff described the discovery as evidence that superagers possess a distinct molecular capability, one that directly enables higher Tamar Geffen, co-director of Northwestern's Super Aging Program and a co-author on the study, put the underlying strangeness of the finding plainly. These are people in their 80s and 90s whose hippocampal tissue still contains immature neurons actively rewiring themselves. A biological signature research teams do not expect to see at that stage of life at all. Beyond neuron counts alone, Lazarov's team also identified meaningful changes in 2 additional cell types working alongside those new neurons. Astrocytes, star-shaped support cells that help regulate the chemical environment surrounding neurons, and CA1 neurons, a specific population of cells within the hippocampus heavily involved in consolidating and recalling memories. Together, these 3 elements, fresh neurons, supportive astrocytes, and resilient CA1 cells, appear to form a kind of coordinated molecular signature, one the researchers describe as a genuine resilience profile, actively helping certain brains cope with the biological wear of aging rather than simply avoiding it through luck. If the idea that some 80-year-old brains are still quietly generating new neurons like a person half their age catches you off guard, you're having the same reaction the research team had, and there's more of this story worth sticking around for. This new genetic and cellular evidence does not stand alone. It slots directly into a much larger body of research. Northwestern's Super Aging program has steadily built since the late 1990s, and together these findings paint an increasingly detailed portrait of what an exceptionally resilient aging brain actually looks like under the microscope. A 2017 study published in JAMA found that superagers' brains physically shrink more slowly over time than the brains of typical older adults, preserving structural volume that most people gradually lose. Later research identified that superagers tend to carry unusually few tau tangles, the twisted protein clumps strongly associated with Alzheimer's-related memory loss, suggesting a kind of biological resistance to one of the disease's key mechanisms. Other work examining the brain's resident immune cells called microglia found that superagers maintain levels of neural inflammation comparable to people 30 or 40 years younger, rather than the elevated inflammatory activity typically seen in aging or diseased brains. Researchers have even found that certain areas of the superager brain associated with attention and motivation appear physically thicker than in cognitively average adults decades younger. Personality and behavioral patterns have emerged as a recurring theme across this research as well. Superagers disproportionately describe themselves as extroverted, socially engaged, and open to new experiences, along with unusually low levels of self-reported neuroticism. Some carry a higher concentration of a specialized nerve cell called the von Economo neuron, a cell type closely linked to social awareness and behavior. None of this proves that an outgoing personality alone can rewire a brain's molecular machinery, but researchers increasingly suspect that a lifetime of social engagement and psychological resilience may interact with, and possibly help reinforce, the same biological pathways. This new genetic research is beginning to map. It is important to be precise here, because it would be easy to walk away from this story believing scientists have located a single, isolated gene responsible for staying mentally sharp after 60. A kind of biological on-off switch anyone could someday test for, or eventually target with a pill. That is not quite what this research shows, and the actual scientists involved have been careful not to claim otherwise. What Lazarov's team has genuinely demonstrated is a distinct molecular and genetic signature, a coordinated pattern involving neurogenesis-related gene activity, specific cellular behavior in astrocytes and CA1 neurons, and elevated production of new neurons, all appearing together specifically within superager brains. This is a meaningfully different, and considerably more useful, kind of discovery than locating one single master gene would be, because it points toward an entire biological system working in concert, rather than one lone genetic switch. The researchers themselves acknowledge real limitations in the current work as well, including a genuinely small sample size of just 6 superager brains and substantial natural variability across human brain tissue more broadly, both of which mean this specific study represents a meaningful first step rather than a final settled conclusion. There is also an important distinction worth holding on to between correlation and cause. The study convincingly shows that this particular molecular signature reliably shows up alongside exceptional cognitive performance in older age. It does not yet fully prove that boosting neurogenesis artificially, say through a future drug or gene therapy, would reliably produce that same exceptional outcome in someone who does not already carry these traits naturally. Untangling that deeper causal question, whether enhancing this biological pathway could genuinely protect an aging brain, or whether it simply reflects some other underlying advantage these individuals happen to share, will require substantially more research across far larger groups of people. Despite those important caveats, it is easy to understand why this discovery has generated real excitement across the aging and neuroscience research community. For decades, the dominant strategy in Alzheimer's and dementia research focused overwhelmingly on identifying and eliminating the damage associated with disease, targeting harmful proteins, reducing inflammation, and slowing structural decline. This research points toward a meaningfully different and complementary strategy: understanding and potentially eventually harnessing whatever biological advantage allows certain brains to actively generate fresh neural material and maintain healthy support cells well into advanced age, rather than simply resisting damage more effectively. Dr. Jennifer Palderai, a memory disorders specialist not involved in the study, described the broader value of super-Ager researcher using a simple, memorable comparison. Every brain, she suggested, is something like a lump of clay. Some people are naturally born with higher quality clay than others, shaped in part by the genetic and molecular factors this new research is beginning to identify. But clay of any quality can still be actively molded, strengthened, and maintained through consistent use, while a neglected lump of clay, regardless of its starting quality, tends to gradually harden and become far more difficult to work with over time. That distinction matters enormously for anyone hoping this discovery might eventually translate into practical advice, rather than remaining purely theoretical. Genetic advantage clearly plays a meaningful role in who becomes a super-Ager, and nobody can simply choose their own underlying molecular starting point. But the same research community studying these 6 extraordinary brains consistently emphasizes that lifestyle factors, physical activity, sustained social engagement, and ongoing mental stimulation appear to meaningfully influence how well any individual brain ages, regardless of their particular genetic starting hand. To understand why the specific discovery landed with such force inside the neuroscience community, it helps to understand just how contentious the underlying question of adult neurogenesis has actually been. For most of the 20th century, the standard teaching in neuroscience held that the human brain was assembled once during early development, and then simply maintained, never expanded, for the rest of a person's life. Neurons, according to that older view, could die off with age or injury, but new ones were not supposed to appear to replace them. That assumption began cracking once researchers in the 1960s and 70s documented genuine neurogenesis occurring in adult rodents and later in adult primates, forcing the field to reconsider its most basic assumptions about how mature brains actually function. Extending that finding conclusively to humans proved to be a far messier, far more contested process. Some research teams reported clear evidence of new neurons forming in the adult human dentate gyrus, a small subregion nested inside the hippocampus. Other teams, using different tissue preparation methods and different detection techniques, reported finding essentially no evidence of ongoing neurogenesis in adult human brains at all. For years, the field genuinely split into competing camps, each publishing results that seemed to directly contradict the other, with no clear resolution in sight. Lazaroff herself has described this exact question: whether the human brain truly continues generating new neurons throughout life, and whether it does so in a way meaningfully similar to what researchers have already established in rodents and primates, as one of the most critical open questions facing her field. The new super-Ager findings do not fully settle that decades-long argument on their own, but they add a genuinely significant new data point, one suggesting that whatever neurogenesis capacity the adult human brain does retain, it appears to vary dramatically and measurably based on a person's cognitive health, rather than remaining fixed or negligible across the board. Beyond its implications for healthy aging generally, this discovery carries real weight for one of medicine's most stubborn and devastating challenges: Alzheimer's disease. For decades, the dominant research strategy has centered almost entirely on identifying and clearing away the damage Alzheimer's causes, targeting amyloid plaques and tau tangles directly, hoping that removing the harmful buildup would restore healthy brain function. That approach has produced some genuine progress, but it has also repeatedly run into a frustrating pattern, where clearing away harmful proteins does not always translate into meaningfully restored memory or cognition for patients. The super-Ager findings suggest a complementary angle worth pursuing alongside that existing approach. Rather than focusing purely on removing damage after it accumulates, researchers may eventually be able to focus on actively supporting or restoring the brain's own capacity for neurogenesis, essentially helping struggling brains regrow some of what they have lost, rather than only defending what remains. Because the newly identified signature involves multiple cooperating cell types, immature neurons, supportive astrocytes, and resilient CA1 cells working together, it also offers several distinct potential targets for future drug development, rather than a single narrow pathway that could prove difficult to safely manipulate on its own. None of this translates into an imminent treatment. Turning a molecular signature, discovered in 6 donated brains, into a safe, effective therapy available to patients typically takes many years of additional research, testing, and clinical trials, And plenty of promising early biological signatures in this field have ultimately failed to produce workable treatments. But for a research area that has seen far more disappointment than breakthrough over the past several decades, a genuinely new, well-supported biological lead like this one represents real, cautiously earned optimism rather than another dead end. Perhaps the most grounding part of this entire story comes not from a laboratory, but from one of the actual superagers participating in Northwestern's ongoing research. Cel Yackley, an 86-year-old Chicagoan and former journalist enrolled in the program, described her own daily routine with a mix of humor and genuine self-awareness. She knits, she exercises regularly, she makes jewelry and sings in a choir, and she deliberately prioritizes staying in touch with friends by phone and video call, even when in-person visits are not always possible. Yackley was careful to note that being a superager does not make her immune to the ordinary friction of an aging memory. Some things she remembers as clearly as if they happened yesterday. Other things genuinely slip away, the same as anyone else's. What seems to set her experience apart is not perfect recall, but a kind of durable cognitive resilience. The same resilience signature researchers are now beginning to trace directly down into her hippocampal tissue at the molecular level. Step back from the specific neurons, the astrocytes and the CA1 cells for a moment, and something genuinely hopeful comes into focus. For most of modern medical history, cognitive decline after 60 was treated as an inevitable, largely unstoppable feature of growing older, a slow fade that better health care could delay only slightly at the margins. This research, and the 25 years of superager data that led up to it, suggests something considerably more optimistic, that certain brains have found a genuine biological path to resisting that decline almost entirely, and that scientists are now, for the first time, beginning to identify the actual molecular signature underlying that path. Nobody has found a single magic gene that guarantees a sharp mind at 90, and any script claiming otherwise would be doing you a disservice. What researchers have found instead is something arguably more valuable at this early stage: real physical proof that an aging brain does not have to slowly go quiet, and a genuine scientific roadmap pointing toward exactly which cells, which molecular signals, and which biological processes future. The story of how long a mind can stay sharp may turn out to be written less in the passage of years, and more in the stubborn, ongoing willingness of a small number of remarkable brains to keep building themselves anew. What began as a simple observation, a handful of elderly patients whose memories refused to fade the way medicine expected, has grown over 25 years into a genuine molecular map, one now detailed enough to point toward specific cells, specific genes, and specific biological processes worth pursuing further. 6 brains will never be the final word on a mystery this large, but they may well turn out to be the first solid foothold researchers have had in a very long time, in the far larger effort to understand exactly what it takes for a human mind to keep its edge, decade after decade, long after the candles on the birthday cake have become genuinely difficult to count.